I'm going to talk about is some of my own work together with some colleagues who I shall mention later and thank. And that is we hope that we have advanced and not set back biological sciences to some extent for societal benefit. But what I would like to highlight is some of the constraints, and I think in applying these advances, and I would like to point that out because I think this is becoming increasingly important and exponentially so. So as a very brief background, I'm sure everyone knows about the advances in medicine and agriculture over the last couple of centuries, which have led to a massive increase in population and a much better quality of life for people. And this includes, as particular examples, the development of, or the characterization of penicillin and the development of high-yielding varieties of cereals. And of course, all of this has been accompanied by an unsustainable use of abiotic and biotic resources of our planet, pollution and climate change, and I'm sure you all know about that. But what I would like to move on to and try and highlight with reference to trying to apply some of our own discoveries is that the competition between population groups for resources and dominance is a situation that is unsustainable and does impact on our own research applications, that I will point out in passing. And secondly, we are quite clear now that the inadequacy of governance mechanisms to be able to resolve emerging societal problems is another feature which occurs at the local, national, and international levels. And of course, this is an unsustainable situation and has to change. Finally, I'd like to mention information distortion in the electronic, print, and broadcast communication media, which is affecting us and our applications, if you like. And the advent of increased use of artificial intelligence is also an issue here. And this is a situation which is unsustainable, and we need to find solutions to overcome it. I mean, this is much easier than global climate change, I think, but it still needs quite a bit of work on our part. So let's look at some of the work that I've been doing in the last 10 years or so. And the first refers to the range expansion of mosquito vectors of arboviral diseases like dengue and Zika, chikungunya, and so on, and of malaria. Now, the predominant arboviral vectors worldwide are Aedes aegypti and Aedes albopictus. And control of these vectors has been highly dependent on reducing the larval sources, sites where eggs are laid and the eggs develop into larvae and then turn into adults. So this is actually quite critical for Aedes mosquitoes. Now, the WHO, World Health Organization, and the Centers for Disease Control in the US guidelines focus on, or only refer to, freshwater habitats of these two vectors. And they also focus on using larvicides like Temifos, which is the most commonly used larvicide for Aedes vectors throughout the world. Anopheles mosquito control, this is the vector of malaria, relies, on the other hand, on insecticides being used against adults, as as being impregnated into med nets, and as sprays applied onto the indoor surfaces of houses and huts. And that is because the Anopheles mosquito vectors tend to bite at night and indoors, whereas the Aedes vectors can bite outdoors and during daytime. So slightly different, significantly different emphasis on control methods. And that's perfectly correct to use by the WHO and CDC. Now, I should also point out that Aedes aegypti and Aedes albopictus have expanded their range from their original habitats in tropical countries, in the tropical area, to semi-tropical and temperate zones. And in fact, there have been epidemics of dengue and chikungunya in Southern Europe and Southeastern Europe. And I believe there have been outbreaks of cases, locally transmitted cases, even in the US. And of course, these are endemic in the Caribbean. Now, what we showed about 10 years ago, and this was observation that was deliberately looked for based on some of early observation of Anopheles mosquitoes, was to see whether the Aedes aegypti species are able to develop in brackish water in coastal areas. And the reason for this is that we did observations in a small peninsula, where any point in the peninsula is not more than 10 kilometers away from the sea. And so this had quite a lot of brackish water habitats for Aedes to our, well, which is what we hoped to see, and we did see. And this is accompanied by biological changes in the mosquitoes that develop in such brackish water and increased resistance to temiphos. Now, brackish water we define as containing between 0.5 to 30 grams per liter of salt. Remember that seawater contains about 30 to 35 grams of salt, and that is considered saline water. So fresh water is anything that has less than 0.5 gram of salt per liter. Now, brackish water habitats have been subsequently found worldwide. We observed that in Borneo Island. It's been observed in the US, in Florida, in Mexico, in India, and Indonesia. And I think this is just the tip of the iceberg. And I think brackish water developing Aedes aegypti and Aedes albopictus are found in coastal areas of many tropical countries, and perhaps in temperate countries as well, in the southern part of the temperate zone. So despite all of this, and this having been reported 10 years ago and in many countries, not only based on our initial observations in a peninsula in the country of Sri Lanka, which is an island, as you know, south of India, but despite all of this, the World Health Organization and Centers for Disease Control guidelines remain unchanged. So as a result, there is no control of Aedes vectors in brackish water habitats along coasts, and such vectors and such habitats form a reservoir, an uncontrolled reservoir of arboviral diseases, which with the advent of rain, the rainy season can spread the disease through freshwater mosquitoes inland and throughout the, but in Sri Lanka, for example, throughout the country. So this is probably much more important than the WHO and the CDC realize, and which is of quite concern. I won't go into the reasons why they don't realize it, but at least in Sri Lanka, the Ministry of Health has now started targeting brackish water habitats for dengue control, for control of Aedes vectors. Let me just show you some examples of the kind of places where Aedes mosquitoes lay eggs and develop into larvae and adults. So A and B are from water collected in in boats and fishing vessels that are sort of found in the coast. C and E are wells that obviously people don't drink the water from such brackish water wells, but they're used for washing fishing nets and so on. And then more widespread, D and F, which are beach debris collections, rubbish collected, collecting brackish water and not just seawater, but brackish water, because it's diluted by rain, in and along coastal areas. Now, what are the biological differences? We can show the upper graph shows you the kind of salt concentrations at which, and this is given in parts per thousand, so 10 parts per thousand is 10 grams per liter. So this is the survival of first instar and third instar larvae of the 2 ED species to adulthood. So it's the whole transformation from L1 to adults. And you can see in the upper graph is their tolerance levels in the Jaffna Peninsula, which I said has quite a lot of salinization, partly due to the coastal nature, and also the increasing salinization of groundwater aquifers due to rising sea levels and still saltwater intrusion. And the lower graph shows you the same 2 species collected from mainland Sri Lanka, that is so less prone to coastal influence. And we note that Albopictus is more resistant to salt than Aedes aegypti, and the third instar more resistant than the first instar, which is what you would expect for other reasons. We showed biological changes. These are transmission electron micrographs of the abdomen cuticle. This is the outer surface, the cuticle of the adult abdomen. So this is fresh water and brackish water. We see a consistently significant increase in thickness, including what we call the exocuticle and the endocuticle. And this is also seen in the larvae. The cuticle is thicker in brackish water specimens than freshwater specimens. But this doesn't seem to hold for the anal papillae, which are osmoregulatory organs, and there is a reason for that we can propose. But so essentially the thicker cuticle, which is found in most of the body of the larvae, which is also surprisingly carried on to the adults. We didn't quite expect that, and that has huge implications as well, which we are looking into. So this thicker cuticle is really one reason why these larvae are more resistant to the larvicide use, that the larvicide doesn't penetrate the cuticle as well in brackish water. Now, does this happen with the Anopheles vectors? In fact, we saw this in Anopheles before we started looking for Aedes. So we saw that in Anopheles culissifaces, which is a freshwater vector of malaria in Sri Lanka. Of course, malaria has now been eradicated in Sri Lanka, so that's not a problem, but it is still an important vector in India and other parts of Asia. Now, Anopheles stevensi is another vector which tends to be a bit more urban than culissifaces. So it has rapidly expanded in the last few decades from northern India to southern India, and then in 2017 to Sri Lanka. And when we try and analyze how this might have occurred, several features. One is adaptation to cement water storage tanks. Secondly, adaptation to develop in brackish water, movement of refugees due to wars, and also the eggs and even larvae can be carried in boats. So these are the factors we think are important, and also windblown across the Indian Park Straits that separate India from Sri Lanka. Very significantly, the Anopheles stevensi has also spread across the Arabian Peninsula, then into the Horn of Africa, and including Ethiopia, Sudan, and now Kenya in East Africa, and Nigeria in West Africa. And that is very recent, within the last year or so. And it's quite clear that movement of refugees and civil wars have been one factor besides the adaptation of stevensi to develop in water storage tanks. And you can imagine that, you know, we have 2 generals fighting in Sudan. We had wars in Ethiopia. And at the end of the day, we have an increase in malaria incidence as a result of the spread of this very potent malaria vector. So it just doesn't make sense, and it's clearly not a sustainable situation. I mean, we really need proper governance in this area. And it is the major defect. We have also shown that larvivorous fish in Sri Lanka, anyway, when you put them into wells, introduce them into wells, they're able to control Stevensite development, but there is a lot of commercial interest in promoting insecticides alone. I mean, we think both have to be used, but fish certainly are a very environmentally friendly method of controlling and an effective method in wells and even water tanks. So this diagram shows you the spread of Anopheles stevensite, which is a major vector which the WHO has taken a lot of notice of. In fact, some of us attended a meeting in Ethiopia less than a month ago that was convened to look at stevensite spread. And so you can see that there's a lot of spread along coastal areas, into Sri Lanka in 2017, Indian Ocean Island, Lakshadweep belonging to India in 2001, and then along the Arabian coast and Iranian coast into the Horn of Africa, and then to East and West Africa now. So this map is a little out of date. And there's no doubt that civil war and the lack of malaria control, mosquito control, was a very serious defect that caused the spread of this vector and increasing malaria in this part of the world. So I'd like to move into another aspect of our work, which involved looking at the relationship between the incidence of dengue and the COVID-19 people movement restrictions, which were quite horrendous, as we all remember, from end of 2019 till 2021, thereabouts, varying between different countries. Now, well-known epidemiologists predicted a collapse of vector control measures during what I shall call the lockdown of movement restrictions, and that this would lead to an increase in mosquito-borne diseases. And this was certainly observed for malaria in Zimbabwe, high incidence during the lockdown. However, we found a very dramatic decrease in dengue incidence in Sri Lanka. And because we had an ongoing parallel project on collection of Aedes larvae from booby traps in a restricted part of Sri Lanka, we were also able to associate this with a much reduced collection of particularly Aedes aegypti, which is the major vector, as well as a subsecondary vector, Aedes albopictus, which was the first time it's ever been shown. And this led us to propose that the decreased incidence of dengue was largely due to the restricted blood meal feeding by Aedes mosquitoes in public places like bus stations, railway stations, places of religious worship, and most importantly, also schools. So if you reduce blood feeding, well-known epidemiological models put forward by MacDonald in the 1950s tells us that the mosquito densities would fall, and this would result in dengue transmission. So all this work has been published and is readily accessible through PubMed. But even what was really surprising to me, and I sat in on this, that the WHO Tropical Diseases Program, even in June, July 2023, continued to emphasize the fact that dengue incidence probably increased. And dengue incidence has been shown to be dramatically decreased, 80 to 90% in Sri Lanka by us, by a few others later, and also in very many other countries now, including South America and some other parts of Southeast Asia. No great change in Singapore, but that's a different story and different epidemiological picture, which we can explain. So the problem here is that most countries rely on WHO guidelines for dengue control and Aedes control. So if the WHO recognizes that the public places are a major transmission hubs, then Aedes control methods can be focused on such places. And failure for the, if WHO does not recommend this, most countries will follow the WHO. Fortunately, in Sri Lanka, we've been able to convince the Ministry of Health to actually target such public places, and they are beginning to do so. But there is a failure here, which I cannot, which is sad. And this is a graph showing you the actual dengue incidence. The green line shows the dengue incidence during the pre-pandemic 5-year period and during the pandemic period. So during the pandemic period, A was the period of severe lockdown. The dengue incidence really decreased dramatically. And this data is for the whole of the country. We have data for every district, including the Jaffna Peninsula, which is what we correlate the density findings with. And then it gradually the dengue incidence begins to go up with the removal of restrictions and then tends to meet the predictions. The predictions are shown in red, and they're based on a very well-known model, the SARIMA model, that predicts the incidence of dengue due to seasonal fluctuations in monsoons. So that's well accepted, and similar results have been demonstrated in Malaysia, for example. So we think it's established, and it's sad that the WHO has not learned the obvious lesson from this yet. So let's move on to another area that we worked on, and that is Lyme disease and tick-borne relapsing fever diagnosis. Now, the Center for Disease Control and Prevention in the US estimates about 50,000 cases of Lyme disease and 25,000 of tick-borne relapsing fever per year in the US. Now, these two diseases are related. They're caused by related groups of Borrelia bacteria, and the vectors are hard and soft ticks. Now, the diagnosis of Lyme disease is heavily reliant on a somewhat ancient two-tier serological test for Lyme disease that was first, after very careful consideration, put forward by the CDC in 1994. There are no serological tests that have been recommended for tick-borne relapsing fever. Now, we have shown, and this has been also known in Europe for actually a decade or so, serological differentiation by line immunoblots using recombinant proteins is quite effective and quite useful in serodiagnosis of Lyme disease. And we've developed similar line blots for tick-borne relapsing fever. And using these, we find that Lyme disease and TBRF are more widespread than presently recognized in the US, and are in fact, co-infections are in fact found in countries like Ukraine and Australia, as well as the US. Now, for Lyme disease, coming back to the importance of that, the different US states now use different criteria. There is very poor scientific communication, and there's, as many of you would know who've been involved in infectious diseases, massive media misinformation, which all makes it difficult to try and put forward tests that are very similar to what's been quite effective in Europe. And I'd just like to show you an example of line immunoblots that we developed. And these are using purified antigens. I won't go into the details of the purified antigens. These are TBRF immunoblots for IgM and IgG. And on the right, we have Lyme immunoblots detecting IgM and IgG antibodies in the sera of patients. So this is the usual this is a useful serological procedure. And so these are the different proteins that I use. C1 and C2 are controls. So essentially, so lane 1, 2, and 3 contain sera that are positive in TBRF. Lane 4 is negative for TBRF and Lyme disease. And lane 5 is only positive for Lyme disease, as shown here. And lane 3 contains a serum that reacts with both TBRF and with Lyme disease. So a mixed infection. Now the kind of inbuilt lethargy to change, if you like, or inbuilt mindset makes it difficult to convince people. And although we've been working with the CDC, it makes it difficult to convince people that here we have a situation where TBRF is quite widespread, there are mixed infections, and we have a potentially important method, which we have refined in conjunction with the CDC for diagnosis of Lyme disease and for tick-borne relaxing fever as well. So lastly, I'd like to move on to discuss COVID-19 vaccines. So at the end of the first year of the pandemic, in December 2020, vaccines became available because platforms that existed, like the adenovirus vector and the mRNA that were being developed for other diseases, were rapidly focused on developing COVID vaccines. So in the very first year, from December 2020 to December 2021, 56% of the global population had received one dose of a COVID vaccine, and most of them had received 2 doses. Now, reliable estimates based on excess mortality deaths suggest that in 185 countries in that 1-year period, 20 million deaths were averted by vaccination. So a staggering number. In the US, the parallel estimate done by independent groups was that there were 8 million fewer infections, 700,000 fewer hospitalizations, 120,000 fewer deaths during a 6-month period from December 2020 to June 2021. Similar data have been published from Israel and probably other countries as well. Now, the critical feature here is that these vaccines were brought in at a time when a lot of people would have died. Normally, COVID causes 20 to 40% of people infected to become severely ill, and many of them to die. So the vaccines were very important. It produced what was called herd immunity. And subsequently, and during that period as well, mild COVID infections in healthy children and adults produce this measure of immunity, which reduces the severity of the disease. So by December 2021, and increasingly so afterwards, because there have been several hundreds of millions of COVID cases who have recovered, we have developed robust herd immunity. The term herd immunity was initially applied in a veterinary context in herds of animals which develop a disease, some survive, and those that survive, immunity. Now, Sir Patrick Vallance, who on TV, speaking with the Prime Minister in his normal, almost weekly COVID address, used the term herd immunity. And he was absolutely polarized by the UK's well-known, rather detestable tabloid press, and almost calling for his resignation. But he was just using a term herd immunity, which is quite well accepted immunologically, had been accepted for decades. Sad, but he survived. So what we have now is an issue to develop better COVID vaccines that can produce better immunity in the upper respiratory tract, which is able to resist variants and is effective in vulnerable populations. And in fact, one of my own observations put forward at that time is that people from tropical areas, with an ancestry in tropical areas, were much more vulnerable to COVID-19 than people during winters, than the people who had a genetic background in the cold temperate zones. So you can imagine, and I propose that hundreds of thousands of years ago, there was a strong selective force exerted by respiratory infections, particularly viruses, as people moved into temperate zone countries, which changed their physiology, as it were, to viruses. So I was also criticized for that, sadly. But I think it's now accepted generally. So what some of the other problems with COVID-19 vaccines? Now, deaths and human misery, we don't want to talk in terms of dollars and cents. Early access to vaccines in some countries was reduced for varying reasons. Some governments delayed vaccination and also were very opaque about their local COVID-19 situation. And there was also incredible resistance to vaccination in many countries due to cultural perceptions and a high volume of misconceptions spread through the print, broadcast, and electronic media. And this is very, it was quite nebulous and quite effective. So misinformation on COVID vaccines may have led to very large number of deaths. And no one is responsible. No one is held responsible, and they should be. Now, misinformation, this spread of misinformation gave rise to what's now called the science, if you like, of infodemiology, or the epidemic of misinformation. And I would like to refer you to this website, which tries to counteract it. These are well-meaning people run from the US initially, but with branches throughout the world, which try work with communities to counteract misinformation about vaccines. Of course, COVID-19 vaccines, the important part of the vaccination phase is, if you like, almost over, and the effects still persist. Because many people who have been vaccinated do get mild infections and develop even better immunity. But we hope that there will be vaccines for the more vulnerable people being developed. So I think that's all I want to say about our own results and how the application, if you like, have been hampered by some of the unsustainable trends in society, which I think has to be addressed. I didn't directly do any work on vaccines, but my daughter ran the Chadox vaccine trial in the UK, and so I was quite familiar with it. And I'd like to also thank my colleagues, Dr. Shah at IGENX in California, and Dr. Surendran in the Department of Zoology, University of Jaffna, Sri Lanka, and other colleagues in both institutions for their dedication to advancing scientific knowledge for societal benefit, despite all these hurdles. Thank you very much, and so I'd be happy to take any questions. Hello, is anyone there?
Thank you so much.
I hope you sort of were able to hear properly. Good.
Wonderful, wonderful talk. And I was wondering, as you go through this and think about mosquito epidemiology, what your views are in terms of climate change, and if there's anything we can do, or is it just, are we just sort of at this point have to accept the fact that the range is going to rise and the seasonality is going to extend?
Well, I think can you hear me now?
Yes.
Yes.
Okay, good. We had fiddled around with the connections I had a bit here. So yeah, I mean, the seasonality of dengue is clearly associated with rains. Rainfall increases the number of mosquitoes, vectors. So that is something that we have to address. But one of the things that we have to do between the peak increases in mosquito densities, we should be targeting the reservoir areas, which are in the coastal brackish water habitats, which until very recently, at least in Sri Lanka, have not been sprayed with larvicides, and there are no instructions to minimize these habitats in coastal areas, like clear up beach debris, make sure water doesn't stagnate in fishing boats and so on. And this applies to all sorts of countries, not just Sri Lanka. But at least we have in Sri Lanka been able to persuade the authorities to take some action. And so once you reduce the reservoir in coastal areas, then you should be better able to control dengue even during the rainy season. One could argue, Singapore, why do you, despite all its control measures, you can ask why there is still dengue in the country. But one reason is close to Malaysia, but secondly, they may have reservoirs in the north which are not targeted. So having said that, so we need international guidelines for this. Having said that, then the other feature is that clearly public places of congregation in countries like Sri Lanka and India, which are not targeted so much in mosquito control, need to be targeted better. Sorry, does that answer your question? It doesn't quite, but I think it highlights what I said again.
Thank you. Yeah.
Yeah.
Yeah. Any further questions, everyone?
This is just Dick Guerrant with an appreciation for your perseverance in presenting this. The brackish water thing, to extend Pranay's question, with the sea level rising, is going to really change the epidemiology of a lot of diseases. I mean, Vibrio bacterial infections in brackish waters are fairly infamous in more temperate climates like Japan and coastal US, and I think most coastal countries around the world. And so this, with the Aedes, all three variants, is really quite frightening and very significant. So I think that going forward, this could be a serious new concern that we've really not anticipated. It's kind of, evolution is changing faster than we realize. Anyway, just a comment to thank you for.
I appreciate that, actually, because I think you hit the nail as you put on the head. I mean, lots of islands, small islands, are very prone to dengue and chikungunya epidemics.
Yeah.
I mean, the Comoros Islands and Madagascar and Indonesia is pretty bad.
Yeah.
Sri Lanka as well. So I think we need to tie all of this together, and we need, and I think we need lead to be taken by organizations that put out these guidelines, like WHO and CDCs look up to quite a lot as well. And they might want to, they've had some very good work showing Aedes aegypti adapting to brackish water in Florida.
Yeah.
Known for about 5-6 years, but it really needs to be taken up. So thank you for that. I mean, we appreciate that.
Yeah. Oh, thank you.
Thank you very much for your time.
Yes.
This was a great talk.
Well, I hope, thank you for listening, and I hope you enjoyed it. Yeah.